This lesson is on stem cells. We have so many things to get through in stem cells, but we are going to focus firstly on mitosis, which is this part of the first statement here. Describe how stem cells originate through the process of mitosis. So let's start with some quick cell number facts, right? You have around 15 trillion cells in your body, and the human body undergoes 10 quadrillion cell divisions in a lifetime. Now, cells replicate in our body for growth of the whole organism, repair, development in children and embryos, asexual reproduction in single-celled organisms. There's so many reasons, and that is why every minute around 300 million cells in your bodies die, because our bodies have the ability to reproduce, essentially. So remember that when cells are dividing, they are actually multiplying, okay? They divide to create more cells, and the parent shares itself by replicating its DNA and sharing its contents between two new daughter cells. So that's the correct terminology for this. Now, every cell in our body goes through some sort of cell cycle, okay? And it spends most of its time in interphase, which is all of that. And it grows, functions, just normal functioning, and it prepares for cell division. And this tiny little extra bit here, that's mitosis, and that is what our focus is. But most of the time, a cell lives in interphase. During S phase, which is part of that interphase, the DNA replication is occurring in preparation for sharing that set of identical instructions to the daughter cells. You'll learn a lot more about this process in Unit 4 in Year 12. Now, mitosis is the phase in the cell cycle where eukaryotic cell nucleus divides, okay? We're not talking the whole cell, we're talking specifically, mitosis is specifically about the nucleus, and it's forming eventually at the end of the process two diploid daughter cells, okay? One parent cell will replicate its DNA and divide and create two identical daughter cells, and both the parent and the daughter cells are considered to be diploid, which means they contain two copies of the chromosomes, right? So one from mum, one from dad, one from mum, one from dad for every single thing. In a human cell, we have 46 chromosomes, so one from mum, one from dad. That's why we have a nice even number. And this is opposed to, say, a haploid cell, which is only carrying one copy of it, which is happening in our sperm and eggs, so that when they combine, they will give you the total 46. Now, prior to the mitotic stage of the cell cycle, the DNA replication is occurring in S phase, and at this stage, the DNA is in a form of a linear chromosome. It's replicated and it remains held together by its centromere, which is that little black dot in the middle, and it's all pinched together. So before replication, the chromosome was only that kind of single ladder. And what we're trying, that's called a single chromatid, and we're trying to get it to have sister chromatids. So two chromatids identical so that we can tear them apart and put them in two different cells, essentially, okay? It's important to recognize that those chromatids on each side, still stuck together, are identical, okay? Because we need identical copies to move into the new daughter cells. Now, mitosis occurs in 4 stages. After the interphase, there is prophase, metaphase, anaphase, and telophase, all following after that. And before the entire cell, or those 4 phases will happen before the entire cell splits into 2, and that's in a process called cytokinesis. Now, obviously, we'll be using these diagrammatic representations of the process because they're easy to understand, but there's so many nice ways you can visualize this under a microscope. So we'll start off with prophase. In prophase, the chromosomes in the nucleus have condensed right down. They're very visible under the microscope, and the two chromatids are still being held together by that centromere. So centrioles are organelles which are going to move to the poles of the cell, so top and bottom, and they will start to form these spindle fibers which will reach out and grab those chromosomes that are lined up in the middle. So the nuclear membrane starts to break down as well, because eventually we want to create a new membrane in 2 new nuclei, I suppose, and we are going to create that spindle fiber network. In metaphase, you're going to see all those chromosomes lining up in the middle, and the spindle fibers have reached out, they're attaching to the centromere of the chromosome, and they're dragged into the center of the cell there. So at this point, it's really easy to observe them under the microscope. In the next phase, anaphase, the spindle fibers contract, they shorten, and they start to pull those chromosomes apart at the centromere, so splitting them in 2 different directions. And the spindle fibers keep shortening and shortening, and they pull the now single chromatids apart, right, towards the opposite end of the cell. And the new daughter cells are receiving one of these, and remembering that these are identical so that they can create identical daughter cells. In the final phase, telophase, this is where a new nuclear membrane begins to form around those chromosomes at each part of the pole. The spindle fibers are breaking down because we don't need them anymore, and the chromosomes become really long and are thinner and much less visible and just really ready to pack into the nucleus. The final stage of cell division involves the rest of the cell splitting too. We've just talked about the nucleus, but we need to also talk and consider all of the organelles as well. So mitosis deals with the nucleus, cytokinesis separates the cytoplasm and takes the organelles with it as well. So cytokinesis marks that point where two new cells are actually created, and it's that remainder of the split. In animal cells, the outer cell membrane will pinch around and will actually close up. But in plant and fungal cells, the cell wall must also be built around the outside of that. Okay, so essentially the cell wall materials are sent to the center of the cell and they're created around there. So those are nice images of that. And so the same thing's happening in fungi. Now this process can take around 2 hours to complete, and certain cells will go through more cell divisions than others, depending on their role in the body. For example, your intestinal cells undergo division every 5 or so days, and they're in constant contact with food, movement and abrasions and things like that. Same with your skin, because they're constantly being bombarded, I guess, with the outside world. Your skin cells can divide every 2 or so weeks, but your nerve cells, there are actually some nerve cells that can stay with you from birth. Now this is quite a beautiful process when it's captured on film, and this is using fluorescence imaging, and it's really easy to visualize at all stages, so just stationary phases down the microscope as well. Look at that, isn't it beautiful? All right, now although mitosis is absolutely necessary, it's possible for the controls to become unregulated. So if an uncontrolled cell growth occurs, it means that the off switch doesn't work. It can lead to cells with mutations, incorrect functions, and they divide uncontrollably. And this is essentially what causes cancers and tumours. So it can disrupt organised tissue, nice normal functioning body tissue, and that means they can also grow all the way down and reach the bloodstream, get access to nutrients, and that encourages their growth. If they reach the bloodstream, they can also snap off little parts of them and start to spread around the body, and that's called metastasizing. So we've really only looked at a very small part of this, but it is so vital to understand the process of mitosis.